Welcome to our educational video on pyrosequencing, a revolutionary DNA sequencing technique. In this video, we will take you on a journey into the incredible world of pyrosequencing, unraveling its secrets and exploring how it has transformed the field of genomics. Pyrosequencing is an extraordinary method that allows scientists to accurately determine the precise order of the building blocks, or nucleotides, within a DNA molecule. With its exceptional accuracy, remarkable speed, and unparalleled versatility, pyrosequencing has reshaped the way we study and understand genetics. What is pyrosequencing? Pyrosequencing is a technique used to determine the DNA sequence of a given sample, enabling scientists to identify genetic variations, mutations, and even pathogens. It relies on the principle of DNA synthesis, where the DNA molecule is amplified and then sequenced in successive steps. The process begins by extracting the DNA from the sample of interest, followed by amplification using a technique called polymerase chain reaction, PCR. This step creates millions of copies of the DNA segment, providing the necessary material for sequencing. How does pyrosequencing work? Pyrosequencing involves a series of enzymatic reactions that occur in the presence of nucleotides, DNA polymerase, and a molecule called luciferase. These reactions generate detectable light signals that are crucial in determining the DNA sequence accurately. As the sequencing reaction progresses, individual nucleotides are incorporated into the growing DNA chain. This incorporation releases a pyrophosphate molecule, which is subsequently transformed into ATP by an enzyme called sulfurylase. The ATP then initiates a reaction with luciferin, catalyzed by luciferase, resulting in the emission of light. By detecting the emitted light, specialized instruments record the sequence of nucleotides being incorporated into the growing DNA chain. The basic steps of pyrosequencing are: 1. Preparation of the DNA template. The DNA template is immobilized on a solid support, such as beads or a microtiter plate, and hybridized with the sequencing primer. 2. Addition of the sequencing reagents. The sequencing reagents include a DNA polymerase, ATP sulfurylase, luciferase, a pyrase, and substrates for the enzymes. The reagents are added in a specific order to the DNA template in a reaction chamber. 3. Detection of the pyrophosphate signal. As the DNA polymerase incorporates a nucleotide into the growing DNA strand, a pyrophosphate molecule is released. The pyrophosphate is converted to ATP by the ATP sulfurylase, which then serves as a substrate for the luciferase. The luciferase produces a light signal that is proportional to the amount of ATP present. The light signal is detected by a charge-coupled device, CCD, camera and recorded as a peak in a pyrogram. 4. Removal of the excess nucleotides and enzymes. The apyrase degrades the excess nucleotides and enzymes in the reaction chamber, allowing for the addition of the next nucleotide. 5. Repetition of steps 2 to 4 until the desired sequence length is achieved. The nucleotides are added one at a time in a predetermined order, such as A, C, G, T. The order of the peaks in the pyrogram corresponds to the order of the nucleotides in the DNA sequence. Advantages of pyrosequencing. Pyrosequencing offers numerous advantages over traditional DNA sequencing methods. Firstly, it enables rapid sequencing, allowing for the analysis of large volumes of DNA in a relatively short period. This speed has significantly contributed to advancing genomic research and clinical applications. Secondly, pyrosequencing provides high levels of accuracy, resulting in reliable DNA sequence data. This accuracy is crucial when identifying genetic variations associated with diseases and understanding complex genomic structures. Furthermore, pyrosequencing is highly versatile, accommodating various DNA regions and applications. It can be applied to whole genome sequencing, targeted gene sequencing, metagenomics, transcriptomics, and many other areas of genetic research. Unlocking the secrets of DNA with pyrosequencing. Pyrosequencing has significantly transformed the way we study DNA, driving breakthrough discoveries in numerous fields. From unraveling the genetic basis of diseases to exploring the diversity of microbial populations, pyrosequencing has become an indispensable tool for researchers worldwide. By deciphering the complex genetic codes, we gain invaluable insights into the fundamental aspects of life and pave the way for personalized medicine and a deeper understanding of our own biology. Narrator, we hope you enjoyed this enlightening journey through the world of pyrosequencing. Remember, this remarkable DNA sequencing method holds the key to unraveling the mysteries of our genetic blueprint. Stay curious, keep exploring, and embrace the wonders of pyrosequencing in your pursuit of scientific knowledge.